Hello, my name is Michael Downey, and it's a great pleasure to welcome you all to the 13th AWRI webinar for this year's program, the first for 2019. This session will take a look at grapevine viruses, particularly leaf roll and grapevine virus A, which can have significant impacts on the economic viability of a vineyard. Now, before I jump over to today's panel, some very quick reminders for the audience. To provide a comment or to ask a question, please open the Q&A section of the webinar, type in your question and click to send. If you'd like to get involved through Twitter, please use the Twitter handle @_AWRI. Also, this webinar is being recorded and will be available to view later this afternoon by the AWRI's YouTube channel. Now, I'd also like to thank and acknowledge Wine Australia for providing funding and support for AWRI webinars. For those of you that have just joined us, welcome. Today's presentation is Monitoring for Grapevine Viruses in Established Vineyards. And I'm very pleased to welcome our two presenters for today. First up, you'll hear from Dr. Nuruddin Habili, a senior research scientist in grapevine virology at the AWRI. Nuruddin has authored more than 150 papers during his 40-year career in virus diagnostics and management research. Our second speaker for today is Dr. Fiona Constable, a research leader for microbiology and senior plant virologist for Crop Health Services Diagnostic Service of Agriculture, Victoria. Fiona has considerable knowledge in epidemiology of viruses and the development of diagnostic tests in various crops, including grapevines. It's fantastic to have both Fiona and Nuruddin, two renowned virologists, here with us today. But before I jump over to Nuruddin, I would also like to introduce Tony Hoare, a senior viticulturist here at the AWRI, who will be hosting today's session and providing some initial background around the topic. Over to you, Tony.
Good morning, everybody, and thank you for your participation in this webinar. I'd just like to provide a brief background about why we came up with this topic. And it is a new format for us to have a panel of two speakers and then a question panel open to both speakers at the end. So I hope it is a useful format for everybody listening today. So why are we looking at monitoring for virus and why are we particularly concerned with this issue? Well, viruses are becoming, it seems, more of an issue, and we're receiving more inquiries through our help desk service. You'll see there is a phone number on the screen and an email address. We encourage you to use us for initial inquiries about virus if you have any suspicions that you may have problems with virus. Hopefully, from the webinar today, you will be able to develop some skills in being able to effectively monitor your vineyards for early signs of virus, and we'll hopefully be able to use pathology efficiently to be able to then get a diagnosis of whether it is virus or not. I guess from our experience here, seeing virus in vineyards, it is becoming, it seems, a more significant issue. And if it is not detected, then it continues to spread. So early detection is very important for the prevention of serious economic damage from virus. Added to that, not only the detection of viral symptoms, which can be confused with other issues such as nutrient deficiencies, trunk diseases, and potential root problems, is also the importance of detecting and monitoring for the vectors of virus. So our speakers will be going into that today as well, looking at how to detect mealybug and scale. It is important, I think, that we develop a protocol for monitoring for virus, and it's going to be a recommendation that we encourage you to at least monitor the vineyards at least once annually, which we think will be hopefully enough, but to be constantly vigilant of both virus-like symptoms and the movement of the vectors through your vineyard. I guess it's really important to look at timing for virus monitoring because the symptoms can be present at certain stages and more obvious to spot. So we'll go into that today. It is also important, depending upon what stage your vineyard's at, as to when you should monitor. We really encourage that if you're contemplating replanting, that it is very important to test for virus in the old material, which you're removing, and also if you're sourcing your own cuttings, the new planting material as well. If you're also top working or field grafting, we encourage that you also test the trunk which you're grafting onto for virus, and also the signwood, especially if you are selecting your own grafting budwood material. So I guess the outcomes we hope are that you will be able to develop a good monitoring protocol to achieve correct diagnosis, and hopefully this will help avoid any costly pathology and allow you to start managing virus if you do have virus. It's also a prelude to a fact sheet which we'll be developing, we're developing at the moment, which is close to release, which will be on GVA Shiraz disease, which has been developed by Nuruddin Habili, and we'll make that available to you electronically after in the next few weeks. I also would encourage you to attend, if you're in the Mildura area, we have an AWRI seminar on May 9th, of which Fiona Constable will be speaking at on virus. So we are including virus as a high priority in our seminar series as well. So there'll be lots of information coming from the AWRI regarding virus. So that's about it from me. I'll hand it over to Nuruddin, who will take you through his talk.
Thanks, Tony. And I start now. I've got only 10 minutes. I have been testing grapevine viruses since 1997, and we were the first to find Shiraz disease in Australia. And when you have a problem related to virus, we would recommend the following. You go and observe the symptoms in the vineyard when you observe them. Either it is autumn, this time of the year, which mostly leaf roll virus, but also it's for the Shiraz and sensitive varieties, other sensitive varieties like Malbec, Merlot. It's also for Shiraz disease, which is related to grapevine virus A. So in grapevine virus A, we have symptoms in the spring as well, not for leaf roll. In the spring, it's standard growth, so retarded growth in the spring. So when you observe symptoms, you record them, vine position, row, panel, etc., GPS. Identify if the vines are infected as clusters, means they are together, infection. That reminds us it might be a vector transmission, because vectors, as we are going to talk about, they are crawlers. So they can only walk from vine to vine, they cannot fly. There are millibags or scales. Or when you observe the symptoms in vines as random, it could be started from the cutting. So it's randomly distributed cutting along the vineyard, and that's human fault. So you position the vines, tag the vines, take photos, and we are happy to help you to see whether it's related to virus or not, the symptom that you have seen. So if it is not virus, it could be something like magnesium deficiency. If you look at these two photos, the top is minus Mg, it means there is deficiency of Mg, and the bottom is leaf roll infection, which the vine got leaf roll. Very similar, except that I am very familiar with the leaf roll, but not with the magnesium. But Tony said that this magnesium is a typical symptom and in the leaf. And when I look at it, it's on the magnesium, you can see the patchy readiness is on and off on the intervenal area. But in the leaf roll infection, it's smooth. But both have got the veins green. So that's the typical for both of them. Symptoms of intervenal reddening, as you can see, and symptoms of LifROL3 and MG both appear at the basal of the, I didn't know that before, Tony told me that both magnesium and LifROL start at the basal position and almost the same time. So it's a bit confusing, but we can work it together. If you send samples to us, we can let you know whether it is a biotic or abiotic symptom. Shiraz disease, I am going to describe. This is the main important, very important disease recently, especially we have seen it in South Australia with grapevine virus A, and it's only 2 countries, South Africa and Australia have it. The rest of the world, they don't have it. And the sensitive varieties, Shiraz, Malbec, and Merlot, but we have tolerant varieties, Cabernet, Grenache, and Nero, the Avola, they don't show up. So if you want to top work on Chardonnay, something with GVA positive, which Chardonnay does not show symptoms at all, you must be taking this in mind that tolerant variety are okay, but sensitive varieties are not. You can see the symptoms, as I said, in the spring, monitoring and observing. In spring, you can see the stunted growth, retarded growth in the one in front, restricted spring growth, and these are the symptoms in the spring. And then you have, as the season goes on, you can see some vine decline here in a 20-year-old wine Shiraz, Trapwork and Chardonnay. It's in Riverland. We have seen this in 2 occasions, in Merlot and in Shiraz, both in the Riverland, South Australia. So we checked for fungus, there wasn't anything there, and we believe that this is GVA-related symptom as well. And this is another, after the season passes, after the harvest, you will see symptoms like very dark purple symptoms, which are typical for, as you can see here, for GVA, Shiraz disease. The leaves curl backward, and the color of the leaves are dark red, and some people suggested it could be phytoplasma. No, it is not phytoplasma, because if you see the cane down the bottom, it's swollen and it's undignified portion, and it's very much like a disease called corky bark with grapevine virus B, not A. So you can see the unlignified wood on the right, and also the even in July, you can see symptoms on the left, which unlignified wood and still some leaves hanging on. Vectors, what transmit these viruses are scale insects and mealybugs. We haven't done much work in Australia about that, but the little work that I have done on scale from Villanga is Partheno Laconium persici. The one on the scale on the left scales, and we crashed the body and we found virus in their body. And on the right, there are mealybugs, which have been known as vectors for a long time. So the grapevine virus A and LifROL viruses, although they are completely different viruses, 2 different families, but they have common vectors, and they are not vector unspecific. That means so far, any scale or any mealybug can transmit any LifROL except ampelovirus, except for LifROL type 2, of course, and 7. We are going to show you more symptoms of other agents rather than LifROL and grapevine virus A, Shiraz disease. Here is a symptom called grapevine yellow speckle viroid, and these viroids are very tiny molecules, that the tiniest that can cause disease. On the left slide, you can see the speckle of the infected leaf showing varroid infection. On the right is Phomopsis, and you can see that, again, like magnesium, here is also confusing. Is it varroid or is it a fungus? You can see that, but easily I can tell that the varroid symptoms are scattered along the veins, but the Phomopsis is scattered everywhere. So that's the difference in the two agent symptoms. Then the another agent is Phytoplasma, which Dr. Fiona Constable is an expert, and we have found these symptoms on Pinot Gris, and there are chlorosis, then necrosis, and when they send samples like that, I take samples from the border between necrotic, the dead tissue, and the live tissue, which shows yellow. That's the most accumulated phytoplasma we can find over there. So these symptoms are rare these days, but sometimes they appear, and if they appear, the vine all gone, no fruit, nothing. Here are the symptoms of grapevine leaf roll disease. You can see both leaves are leaf roll infection, but you can see the pattern of the infection a little bit different from them, each of them. And leaf roll virus type 1, this one is a Langhorne Creek nursery, which shows infection just started. And these are the symptoms of some unusual grafting compatibility, which we found this recently. They come from Tasmania, and you can see the leaves that they are not green vein; they're just uniformly red, and it does not look like it's a GVA or Shiraz disease, but we have these problems before. We couldn't find any virus in them, but one of the growers, very famous, with these symptoms, with grafting compatibility, I found that, what happened? They said it's got over it. I said, how? They said because the first year cropping too high, too much, they couldn't get enough food, and that's why they're swollen on the top. And once the crop load was low, they got over it. So this is another problem. We don't know what it is. The detection techniques, what we use, we do PCR on top, and you can see the positives as the white dots, and ELISA down the bottom, it's yellow and colorless symptoms. Yellow ones are the positive. And the final slide, well, what we do here, we eliminate viruses. It's a year-long, at least, business of eliminating viruses from grapevine, starting from a few cells until we grow the cells under thermotherapy and chemotherapy. And after a year, they go into potted soil, and then they given to the client, and the client, we want them, another 2 years dormancy testing before being completely released. So thank you very much for listening. If you have any questions, I'm ready to answer.
We might hold the questions until after Fiona's talk, and just have, we're getting a few questions coming in. So we might wait until Fiona's presented her talk, and then we'll have time for questions at the end. So thank you, Fiona. I'll hand it over to you.
Yes, so thanks very much for inviting me to talk today. I'm just going to work through, I'll probably reiterate some of the points that have already been made in terms of viruses. I've particularly got to focus on leaf roll and GVA for this session. So when you're monitoring for leaf roll viruses, I just wanted to show the range of symptoms that you might observe. These symptoms tend to appear late in summer and in autumn. On the left-hand side, you can see leaf roll 1 infecting Marzamino and Pinot Gris. So you're still seeing those rolling leaves, but not necessarily strong reddening or interveinal sort of greening. And at the bottom on the left-hand side is Pinot Noir infected with leaf roll 1. But on the right-hand side, there's also Pinot Noir, and it's infected with leaf roll 1 and 9. And so I guess this demonstrates that perhaps one thing that can happen is that once you get mixed infections, the symptomology might be exacerbated. Symptom expression certainly does depend on virus species and strain, and leaf roll 1 and leaf roll 3 are often thought to be more severe than the leaf roll 4 viruses. But we also see an impact through grapevine varieties as well, and some varieties, as Nuruddin explained earlier, are more tolerant to infection. One thing that we've observed over time is the impact of environmental conditions. So we know that extreme temperature, for example, can impact leaf roll expression and extreme heat. So it's important to keep that in mind when you're monitoring for your vineyards. Certainly symptomless infections occur, and so it is important to make sure that you do maybe undertake some active testing at times to ensure that that vineyard is in fact not infected with viruses. The other thing that we've observed is that after an infection event, it can take quite some months before we start to see symptoms. It can be greater than 12 months and sometimes even longer. As Nuruddin pointed out, there's lots of other factors that cause similar symptoms, and sometimes if you're not as familiar with the symptoms as someone like Nuruddin, it's important to just double check. We certainly get a lot of calls through summer around the reddening that people observe, which may sometimes be associated to drought, particularly if you see those symptoms on the basal leaves of a grapevine. And as I say, symptoms should be confirmed by a lab test. For GVA, I think symptomology is not as straightforward. Certainly some grapevines express Strauss disease that Nuruddin explained, that reddening, but there are plenty of grapevine varieties out there where you may not see symptoms. But there still may be an impact. Sometimes viruses can have a more insidious effect, where we're really seeing an impact on quality and yield, even if not specific symptoms. And this might be because there's been an impact on graft take and damage to the vascular system that doesn't allow the plants to grow as well as they should. So again, if you think you have a problem with virus, a laboratory test is very important. Virus infection is definitely for life. Once a grapevine is infected in the field, they stay infected. And viruses get transmitted in a number of ways. So certainly insect vectors are very important, and I'll talk a little bit more about that in a minute. But they're also transmitted through vegetative propagation. So you have to test before you want to graft or top work your vineyard so that you're not perpetuating disease and further impact down the track, either from the signwood that you graft on top or to the rootstock that in itself may be infected but have symptomless infections. So I highly recommend getting high health planting material through vine improvements and certification programs. There's quite a few vine improvements in each state. And you want to do that because the impact that viruses have on the vigour of your vineyard, the longevity of your vineyard, and the yield and fruit quality. The wise words in red down the bottom are from my old PhD supervisor, Bob Simons, who believed that your grapevines were the most important investment in your vineyard because they're the things that produce the grapes that you know, keep you going. Infected vines are certainly a source for virus for insect vector transmission, and mealybug and scale have been reported to transmit GVA, GVB, leaf rolls 1, 3 and 4, including strains 5 and 9. So it's really important to be on top of mealybug and scale in the vineyard. The greater the population, the greater the risk, because you have more insects simply to transmit the viruses. We seem to be having a greater problem with grapevine and frosted scale in Australia. There is some evidence that these do transmit viruses, and so it's really recommended to keep on top of these, but a lot of the mealybugs are also problematic as well. When mealybugs and scale acquire a virus, it's what we call semi-persistent. That is that they acquire virus and then they can retain that virus for a short period. It could be several days, and then eventually they're uninfected, but that doesn't stop them from reacquiring the virus. When they do acquire the virus for transmission, there may be no latent period. That is, they can acquire the virus and then after a short time frame they can transmit it. So it's really important to keep that in mind in terms of control. The transmission can be quite quick. What we find, or what's reported around the world, is that the crawlers, the nymphs, are much more efficient vectors than the adults. It's probably because they are moving. So again, if you start to see mealybug and scale booming in your vineyards, it's important to try and control those. And on their own, mealybug and scale can also cause damage. So there's another reason to retain control of these insects. This is an example of a vineyard that we visited last year. The grower had observed increasing problems in his vine in the vineyard, and you can see on the bottom left-hand corner a grapevine that was impacted by multiple viruses, in fact. And then next to that, some of the symptoms we were observing that were typical of leaf roll. So he had leaf roll 1, leaf roll 3, leaf roll 4 and GBA, all transmitted by mealybug, and you can see the scale on the left-hand of the crawlers on the left-hand side. And in the middle of the scale. So when you have a mealybug outbreak, they are active and they can crawl from vine to vine, and if they're infectious, they can crawl vine to vine and take an infection with them. So you get within row vine to vine transmission. But the crawlers in particular can also be dispersed on wind and result in random infections within the same block and between blocks and in vineyards. The other important aspect is human-assisted movement. So again, that can lead to spread along the vineyard and localised infection within and between vineyards, and that can occur on machinery, so harvesting equipment and the like. It can occur on people's clothes and shoes. So it's, again, really important to retain control of insects to slow down that spread. When you're managing your infected vineyards, there's probably a few questions that you need to ask yourself. Diagnosis is really important so that then you can be provided with enough information to make those decisions about how you're going to manage the disease. It's important to have a virus infection accurately diagnosed. On the other hand, it's also important just to manage the vineyard health so that you don't see so that you can manage the overall quality of the vineyard, including nutrition, pests, other diseases that might also impact or contribute to problems with virus, and just to make sure that it's not actually those factors that are causing the problem. If your vines are infected and seem okay, well, that's fine. Some virus varieties may not be seriously affected. Some are, may be symptomless, but they can be a risk to neighbouring vineyards because you can get that transmission from vine to vine through insect vectors. On the other hand, even though you think you may not have an obvious impact, you're not seeing those bright red symptoms on some of your vines, it's probably important to assess the vine performance, fruit quality and yield to make sure that they're actually not having a more, as I say, insidious impact that leads to the loss of the sustainability of the vineyard, the productivity of the vineyard. Your options to control GBA and leaf roll are several. First of all, it's important to implement good hygiene to control spread, so you control mealybugs and scale. You consider the movement between vineyards with equipment and people so that you're not carrying infectious vectors with you. So start clean and then move to infected areas. And clean down machinery between vineyards as well, so that you don't take those viruliferaous insects with you. A practice that's been really useful in New Zealand for low-level virus and disease incidents is to remove affected vines. As I say, grapevines are a source of infection for vineyards. So by removing those vines, you take away the virus and the risk of transmission. It's also important to test the neighbouring vines because of that vine-to-vine spread. Even if they're symptomless and if they're infected, you should remove those as well. And certainly monitoring regularly, at least once a year when symptoms are probably going to be obvious, and actively test in areas where you've seen disease in the past is really useful. Once you have a high level virus and disease incidents, you might want to consider whether you can live with it or whether it's viable to replace the vines in the entire vineyard. These are economic decisions that only you can make as a grower. As I say, vineyards can be a source of infection for other vineyards. So if you do retain that vineyard, you need to control spread to minimise the impact. And you do need to assess what the risk is to other blocks and vineyards as well. So it may well be that economically it is better to remove the vineyard than to retain it. From a quality perspective, but it may also be, if you can manage spread, that perhaps your decision is that you leave that vineyard there, noting that they are a risk to other vineyards. Virus protection is really important. Symptom expression isn't a reliable indicator of virus infection because you have those lookalike diseases and symptomless infections, and it's really important to use accurate diagnostic tests to verify virus infection. And you can get testing done at AWRI with Nuriden or with our lab services here in Victoria. And I think that's it for me.
Thank you, Fiona. We've received quite a number of questions, so we might go straight to the questions to keep the time under control. So the first question is to both speakers. Where is the best place or places on the vine to take lignified cane samples? I understand that the titer may be higher nearer the trunk and lower on the cane. Would one of you like to speak first?
We usually recommend that people provide us with at least 2 shoots per grapevine, and that they give us the basal parts closer to the trunk, and we usually ask for 20 to 30 centimetres from which we can take a sample. And we usually like the leaves attached.
Thanks, Fiona. Nuriden, would you like to add to that?
Not really. That's what we believe in for viruses, but for phytoplasmas, we take leaves. You know, it depends on for the Agrobacterium crankle, we get the gall tested. So it depends on the pathogen.
Yeah, other pathogens may require different sampling processes.
Yeah.
So I guess to add to that, potentially it may be best to send photos or images through first of suspected symptoms, and then the various vegetative parts can be sent in for pathology once it's been narrowed down as to what the problem may be from the photos. Is that a fair suggestion?
Yeah, I mentioned that in my slide.
Yeah, all right. Second question is, where is it best on the vine to take green tissue samples?
It's probably a pretty similar process. In fact, again, at a minimum of 2 shoots per vine, and we would probably prefer to have the more basal parts than nearer to the trunk or the cordon than to take the shoot tips.
Yeah, better to avoid very tips of the shoots.
Yeah.
Avoid tips. Okay.
Yeah.
Third question, explain the pros and cons of ELISA versus PCR.
Okay, ELISA is limited. We don't trust many commercial kits, and what we do is only using Lifro 3 for ELISA. Even GVA is not available. it's there, but we don't trust them. So ELISA, that's it. And for PCR, we can go for whatever viruses they are, and we can make primers. It should be known viruses, sequenced viruses, not unknown ones. But there is another new system called next generation sequencing, which they can detect even unknown viruses.
That's true. The benefit as well with PCR is the greater sensitivity. So it allows a greater pooling of samples for diagnostic testing, particularly if the virus concentration in the shoot is low. The way that it works is that ELISA captures only a certain number of viruses, but with PCR, we're actually able to make exponentially multiple copies of the target region of the genome of the virus to a point where we should be able to detect it. Having said that, no test is 100%, and there will be times when viruses are present but below detectable levels.
Thanks, Fiona. Next question 4. What is the ideal time of day to take samples for virus testing? And 2-part question, does temperature at the time of sampling have an effect on the pathology results for virus?
Oh, yeah, I believe yes. We tried Pinot Gris virus during the hottest month of the year versus the cooler month. Then the hottest month was a very low detectability, while in the cooler month was a much better detection. We always believe the viruses don't like hot weather, but this is reverse for viroids. Viroids love hot weather. It's all depends on the type of pathogen, and but we recommend cooler time of the day.
Yeah, morning or late in the day, and keep samples cool before sending them. If they get cooked, by the time they reach the lab, they can be in a state that makes it difficult for us to be able to test for viruses.
Yes.
Thank you.
We also ask that people send samples early in the week, Monday to Wednesday rather than Thursday, Friday, so that samples don't get sort of stuck in the post over the weekend.
Sure.
Thank you. I'll have a question from Libby. The industry, in particular vine improvement, really needs one consistent protocol across labs for sampling from individual vines in the vineyard for virus to reduce the risk of false negatives. The recent sampling project for GPGV found that sampling 10 leaves from one vine and testing them individually resulted in 9 positives but 1 negative. Can a consistent low-risk protocol be agreed upon between labs and communicated to industry?
Good idea.
Yeah, so that was a really nice little piece of work, and it did show that we could use that protocol for GPGV. It would be nice to prove that also for the other viruses. 10 leaves actually might be easier to sample and give a greater representation of the grapevine than just 1 or 2 shoots. So it'd be really nice to verify that.
Yeah, Pinot Gris virus, leaves with wet stems and canes for different viruses, and GBA. Leaves we don't use much unless the viruses are only limited to the veins. So how come we get the leaves, and they rotten very quickly during hot temperature?
And we were thinking about using the base of the petioles rather than going into the actual leaf blade.
Oh, okay. I thought it would be leaf, yeah.
I'll let you two discuss that a bit more, and we'll provide advice back to Libby once we've heard back from a discussion that you two might want to have afterwards, if that's okay, because we've got a few more questions. I've got a question here from Alejandra. I have a question about virus transmission. Would pruning gear spread the leaf roll virus moving from an infected vine block to a non-infected vine block? Yeah,
no. These viruses are generally not mechanically transmissible, so pruning equipment is usually not a risk
But I would like to add that pruning equipment, if you start with the infected area, they might carry sticky mealybugs.
Yeah.
And you don't wipe them out, the mealybugs will go to the non-infected and they establish themselves and they might infect. So it's an indirect way of transmission.
Yes, that's a really important point.
Yeah. Thanks, Nuruddin. Thanks for that. I have a question from Gerardo. Have you seen some effect of elicitors to decrease the effect of leaf roll virus?
I haven't personally, no, not personally.
Nuruddin?
No.
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 Question from Robin. Is virus expression increased in hot conditions or decreased? I think we've answered that, but we'll go into maybe a bit more detail. Do you see lesser or greater virus symptom expressions in warm inland areas?
Yeah.
Do you mind if I go first, Nuruddin? So
You go first.
we did a trial a few years back where we compared testing and symptom expression in a cool climate, which was in the Yarra Valley, versus a Sunraysia area and around Loxton. And actually we didn't see terribly different expression between the two viruses in each year, but as I said, we saw similar symptom expression in each year in each region, but those extreme weather conditions, which were actually around the 2009 bushfires here in Victoria, and a large portion of the southern part of the country experienced those, was when we didn't see symptom expression in 1 year when we anticipated to see it.
Yeah, the Swiss thought about eliminating viruses using hot climate of Tunisia, and they planted Shiraz vines over there. We infected with virus, and in 40 degrees, they didn't show up symptoms, and they took the tip and replanted and got rid of the virus, at least by ELISA testing. So in hot weather, that showed that the virus does not like to replicate, and it could be that it's in global warming would help reduce the virus. Oh, that's one thinking, sorry.
Yeah, well, I guess, Nuruddin, that's the premise of heat treatment for virus eradication, to expose grapevines at high temperatures, or any planting. You can do this with other plants as well, for a period of time. At that point, the virus replication slows down and the shoot tips can grow away from the point of infection. And then if you take those shoot tips and those on, then you may see virus-free plants.
Yeah.
Thank you. We have a question from Hugh. Are nematodes and root-feeding mealybugs important as vectors?
Yes. Yes, nematode for fan leaf, isn't it?
Yeah.
For Nipah viruses, but we don't have here much. And root feeding is in New Zealand. Yeah, up to down to 35 centimeter deep in this. Well, they live in New Zealand, and they found that very important.
Same in South Africa as well.
And what about nematodes?
Nematodes fan leaf in Europe, very important. It's the Finema index. It's a fan leaf vector. We don't need to worry about it. We've been testing for fan leaf. We couldn't find any.
Yeah, fan leaf isn't known to be present in Australia, and it's probably it was found at one point in the Rutherglen area and somewhere else, but it's believed to be eradicated.
So that's a specific relationship between a species of nematode and fan leaf virus. So
that's correct.
They're not generalist vectors of viruses. There's specific relationships between.
No, not at all.
Yeah.
Okay. Gerardo has a question. Is the Shiraz or Syrah decline related to some virus, or is there a genetic cause?
Oh yeah, that's a good question. And the Syrah decline is different from Shiraz disease. It's GVA related. Syrah decline recently, they in French and Californians believe that it's a genetically related phenomenon. So needs more work to be done on it.
Anything to add, Fiona?
No, I agree with that point. The Europeans and the Americans say genetic for Syrah decline.
Thank you. Pete has asked, what are the control measures for mealybug and scale? I might answer that question directly to Peter because we have some, but I can direct everybody to the AWRI fact sheets, which are on the website, and there are there's a lot of information there on mealybug and scale, and also Wine Australia on their website have a lot of information as well. But I'll direct that directly. I'll answer that directly with Pete. Martin asks, controlling virus vectors is certainly a key factor, but mealybugs and scales are notoriously difficult to control with pesticides. What strategy do you recommend? Monitoring thresholds, timing, type of chemicals. Once again, there is a lot of information on controlling these vectors on the websites of AWRI and Wine Australia. So I encourage Martin to have a look at that. And also, if there are some further questions, Martin, if you'd like to just contact the help desk directly. Leah asks, what sample is required to test for leaf roll virus?
Fiona, you can.
Yeah, so as we said before, we have found that you can detect leaf roll viruses at most time of the year, whether it's green tissue or dormant tissue. And you would probably want to take the basal parts of the shoots with leaves attached if they're present, and 20 to 30 centimetres and at least 2 shoots per vine.
Yep. And dormant canes also suitable.
Yeah.
Thank you. Gerardo has another question. Is there a special management to make before replanting vines on some area with high infection in previous vines?
Yeah. So I think it's important to try and remove as much of the previous vine or vineyard before replanting, because vines are notoriously hard to kill and they will reshoot, and they may harbour vectors and virus that will then be transmitted to the new vineyard. So it's critical to remove as best you can that material and try and control vectors when you do replant, just in case you don't remove as much as you can.
Yep.
Richard has a question for Fiona. You mentioned extreme weather can influence symptoms. What kind of influence can this be?
So with high temperature, we observed that symptom expression of leaf rolls, we just didn't see the symptoms in that particular year, and it seemed to coincide with high temperature events. So again, it might be that the virus is still present, but maybe it's not interacting and replicating as much as it normally would. It would be really nice to measure that. We haven't done that. But it may be that it's associated with replication of the virus and how it interacts with the host for symptom expression.
Mauricio asks, is there any product that can be applied to plants to minimise the virus damage in plants?
At the moment, I would say no. There's work being done on interfering RNAs as a potential control, but I think at the current time, no, there's nothing you can apply to minimise virus damage. You really just need to, if you have a virus infection, I'd encourage just to manage that vineyard really well to improve all the other aspects.
Richard asks a similar question. which we've just had an answer on, but I'll repeat it. Practically speaking, if a vine is removed due to a virus, can a new vine be planted in that space, or should it be left for a period of time? Reason being, could remnant infected roots pass on the virus to the plant?
Yeah, so as I mentioned earlier, if there's material left in the ground, the risk is, as long as it's alive, the potential is that if any vectors or mealybug or whatever are subterranean, the risk is that they could acquire the virus and transmit it.
We had a trial in Barossa Valley for Pinot Noir when we removed the infected vines and the healthy vines together, and we planted 7 months later in the holes, the same holes, infected and healthy. The vines planted in the infected holes, they got back the virus, but the vines who planted in the healthy area, they didn't have the virus, so it was negative. So that exactly says that, that was 7 months, but I heard that in New Zealand, even 4 years after vacancy, the land, without any plant, they come back, and that's terrible.
Thank you. Leah asks, is non-lignification of shoots on an otherwise healthy-looking cane always a symptom of a virus? Is there more than one virus causing this symptom?
Always.
No, I think lack of lignification probably has multiple causes. Phytoplasms are certainly something that contributes to that as well. Just because you see no lignification, there could be other abiotic or biotic causes. So it is important to get virus testing done if you suspect virus.
Thanks, Fiona. I think that's answered it. Leah also asks, is there any likelihood of applaud being registered for scale insect control? Currently only registered for mealybugs. Leah, I'll take that question up with Marcel, and we'll get back to you directly on that one.
Yes.
Robin asks, considering that healthy planting material is the most important investment when establishing a vineyard, do you think that virus management should be included in Australia's grapevine nursery standard, similar to New Zealand grafter grapevine standard?
Definitely.
Yes, we can't disagree there, Robin, and I think that's something that probably needs to be addressed at more of a policy level. But yes, it should be included for sure. I've got a question here from Alexandra. In an organic vineyard, what are the best ways to control mealybugs? There are some options there for biocontrols, and I can discuss them directly with Alexandra afterwards. So I'll take that question and get back to Alexandra directly there. Michael asks, in cooler, wetter seasons, we tend to see symptoms of leaf roll earlier and on a greater proportion of leaves. If removing vines, we try to allow 2 years before replanting so that old root fragments are less likely to be a source of virus. I'm not sure what the question... if removing vines, they try and allow 2 years before replanting.
2 years.
I think there may be a question there is what is the lag phase between removing vines and replanting? Is there a period that we can recommend, or is it really to do with the degree of removal of material?
I gave an example about New Zealand. 4 years later, still come back.
Yeah.
So
I think, that would be something.
We haven't done any work here.
Yeah, I was about to say exactly that. It's something we probably need to investigate here. Our vector transmission, which really needs to be proven properly with mealybug and scale, we need to do work in that space, and we need to understand also how long you need to leave something fallow before you replant. We just don't know the answer to that question for Australia.
Thank you. We have one last question from Ben. Hi, when replanting a vineyard, would planting a green manure crop like mustard and then disking into soil prior to replant help to fumigate the soil and get rid of remnant mealybugs in the soil? I could probably answer that to some degree, Ben. We have heard from a grower in the McLaren Vale region that planting a crop of beans caused a massive explosion in the population of mealybugs in that vineyard. So as far as planting, I think you do need to be careful which crop you do select and whether it can actually enhance the breeding cycle of a vector such as mealybug, which was found to be the case with beans in this vineyard. They also then experienced a great increase in virus symptoms throughout the vineyard after that as well. So I know that from previous experience that there is an association between mustards and fumigation with nematodes. It does have an antagonistic effect on nematode populations in soil when it is worked into soil, but I'm not sure about mealybugs, and I'll throw it to Nuruddin and Fiona.
I don't know the answer to that question.
Okay.
We can do a literature search for you, Ben, and we can get back to you and see if there has been any work done in that area.
Yeah, good idea.
That's all for the questions. So I'll just thank you all for listening in, and I'll hand it over to Michael just to wrap things up.
Okay, fantastic. Thanks very much, Tony, Fiona, and Nuruddin for providing the content for what's been a really useful session. Clearly a lot of questions out there, so hopefully if you did have some questions, you've found some answers here. If you haven't, the AWRI's help desk contact details are there, and that's probably a good first place of contact for any queries you have. So yeah, please do contact the help desk. I'd also like to thank the audience for participating in today's session. All attendees will receive a follow-up email with a link to the recording of this session. The AWRI will be hosting a couple of webinars next month, so please stay tuned for more on that. So thank you again for participating, and I look forward to seeing you again at the next AWRI webinar.